Hello, my name is Joshua Mugabe. I welcome you to Chemicals of Life. All living organisms are made up of chemical substances. So when we are talking about living organisms, we say all of them are made up of chemical substances. And those chemical substances, they can be lipids, they can be proteins, they can be glucose, fats and oils, and many other. Reactions between these substances keep the cytoplasm and the organism alive. They are living processes. They are reactions that take place in the cytoplasm. And these reactions, we call them the living processes. And they are the ones that keeps the organism alive. We have reactions or processes like excretion, respiration, osmoregulation, temperature regulation. All those are physiological processes, and they are living processes that keep the organisms alive. The chemical substances described in the next series of slides are carbohydrates, proteins, and lipids, but there are 100 of others. So these chemical substances that we are mainly going to look at, we shall look at the carbohydrates, we shall look at the proteins and the lipids, but there are very many other chemical substances that are used in the body or that are carried out in the cell of a living organism. Carbohydrates contain elements carbon, hydrogen, and oxygen. So these chemicals, they are made up of units which we call elements, and these elements for the carbohydrates, we have carbon, we have hydrogen, and we have oxygen. So when you look at our structure here, this is glucose. Glucose is a carbohydrate, and you can see that it is made up of carbon. You can see there is carbon here, have carbon, a lot of carbon elements. You can see a lot of carbon elements in many places. It is made up of hydrogen. We can see hydrogen elements. There are very many of them. Hydrogen, and this is glucose, alpha glucose, and alpha glucose is a carbohydrate. And then they are also made up of oxygen. They are made up of oxygen, and we can see a lot of oxygen elements which are in the carbohydrates. So the carbohydrates are made up of elements which are carbon, hydrogen, and oxygen. Familiar carbohydrates are the sugars and starch. Those are the most common carbohydrates. We have sugars, like for example here we have, this one is glucose, we have fructose, we have maltose, all those ones are sugar, and many others. And then we also have starch. Glucose, we have fructose, we have maltose, we have sucrose. All these ones are sugars. Those are some of the carbohydrate sugars that we have. Glucose and fructose have the same formula, which is C6H12O6. That is glucose and fructose. They have a formula of 6 carbon atoms. Hydrogen, they are 12, and oxygen, they are 6. So when you look here and you try to look at the carbon, let us try to see the carbons here. We have this one is a carbon, that is 1. We have this one is a carbon, those are 2. Here we have another carbon, those are 3. We have another carbon here, these are 4. We have another one here, those are 5. And we have another one here, those are 6. So you can see here that this one is correct. We have 6 carbon atom. So it will be the same when you go, for example, to count hydrogen. You'll find there are 12. Oxygen, you are going to find there are 6. Let us try to count oxygen and we see whether there are 6. We have this one here, this is 1. We have another one here, these are 2. We have another one here, these are 3. We have another one here, these are 4. We have another one here, these are 5. And then another one here, these are 6. So you will find that glucose and fructose, they have the same formula, which is C6H12O6. 6 carbon, 12 hydrogen, and 6 oxygen elements. Sucrose and maltose have the same formula, which is C12H22O11. That is sucrose and maltose. They have a general formula, which is 12 carbon, they have 22 hydrogen elements, and then they have 11 oxygen. Let us try to prove out and see. This is sucrose. Let us try to see if it is true. We can get carbon. Carbon, we have this one is 1. There is a carbon atom here. This is 2. There's another carbon atom here. Those are 3. Another one here. These ones are 4. This is 5. We have another one here. Those are 6. We have another one here. Those are 7. Have another one here. Those are 8. Have another one here. These are 10. Have another one here. These are 11. Have another one here. These are 12. And then we have this one here. These are 11. These are 12. So when you count, you will see that they are 12. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there are 12 carbon atoms or carbon elements. So even when you count hydrogen, you will find that there are 22, and then oxygen, you will find that they are 11. Let us just try to count oxygen. So oxygen, we have this one is 1, this one is 2, we have this one is 3, we have this one is 4, have this one is 5, we have this one is 6, have this one is 7, have this one is 8, have this one is 9, have this one is 10, saying 10. But we say there are how many? 11. Where is the problem? Is there a problem? There is one here we have not counted. Somewhere here, this one is 11. So when you count them, you will see that there are 11. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 oxygen atoms. So sucrose and maltose, they have the same formula, which is C12H22O22. That means 12 carbon atoms, 22 hydrogen atoms, and 11 oxygen atoms. Carbohydrates, these ones provide the main source of energy for respiration in living organisms. So all the living organisms, the major source of energy is the carbohydrates. They are the major source of energy as carbohydrates, and that energy can be used in respiration. Glucose and fructose occur in fruits like grapes. So glucose and fructose can be found in fruits like grapes. That's where we find glucose and fructose. Sucrose is familiar to table sugar and comes from sugar beet or sugar cane. So sucrose is the same as the sugar we take in tea, or what we call sugar cane sugar or sugar beet sugar, or it is the table sugar that we use in our tea, that is sucrose. Maltose is produced by germinating barley and is also the sugar used in brewing beer. So maltose is another sugar like sucrose. This one can be produced by germinating barley and is a sugar that is used in brewing beer. Note that the names of the sugars ends with OSE. So most of the sugars, we give them the name of OSE. A glucose molecule has a straight chain. A glucose molecule has a straight chain. Now let us try to look at the glucose molecule and try to understand how it looks like. It has a straight chain. So we have carbon connected to carbon to form a straight chain, and we have 6 carbons. If you look at them, this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and the 6th carbon. So there are 6 carbons that make up a chain, and they are connected to one another by a bond. We have a bond connecting one to another. That one is a bond, a bond, a bond. We have a bond, and then other bonds will connect to oxygen and hydrogen atoms. So carbon has 4 bonds. Carbon is supposed to have 4 bonds. Now let us try to join the other bonds, and we see If we go on joining those other bonds, that's what we are going to get. So here we shall have our carbon, then we have OH, and then we have here carbon, OH, H, carbon, we have here carbon, OH, H, H, OH, H, OH, and then here we have H2OH. So that is how our carbon looks, or glucose looks like. It has a straight chain, and this is our straight chain of glucose. Five of the carbon atoms may be arranged in the ring. 5 of the carbon atoms may be arranged in a ring. We can arrange 5 of the carbon atoms in a ring. And how does it look like? We can put like that. So 5 of them, of the carbons, we are going to put them in a ring. So if you look here, this is our ring. We have our ring here, and it is made up of 5 carbons. We have this one is 1, 2, 3, 4, and then 5. So 5 of them are forming a ring. This molecule is often represented by simply as an hexagon. This molecule of glucose molecule is often represented as an hexagon. If you look here, we have said that we have arranged it in a ring. That ring is hexagonal. So when you look at this hexagonal, it is representing this molecule of glucose. Two molecules of glucose can join together to form a molecule of maltose. So I want us to look at this diagram down, and we look. This one is a molecule of glucose, what you are seeing here. And this is alpha glucose. Even this one is a molecule of glucose, and it is also alpha glucose. So what is going to happen is that one glucose will use carbon 1, and the next glucose will use carbon 4. So carbon 4, if you look at this glucose here, let us look at this glucose. This is carbon 1, this is carbon 2, here we have carbon 3, and this is carbon 4. So this one is having carbon 1. Now when you place it here, it will be carbon 1 here, carbon 2, carbon 3, carbon 4. So when they are going to join this one and this one, carbon 1 is facing carbon 4 of this one. So this one has got a hydroxyl here, hydroxide here, hydroxyl, and here it has OH and OH. So these two are going to combine, and we shall lose a water molecule from here. We can get this OH and one H here. So what we are going to remove, we are going to remove this OH and this H here. These are the ones that we are going to remove, the OH and this H here. So when we remove these ones, they are going to give us a water molecule. We shall get a water molecule, meaning that this bond here is going to be broken. So when this bond is broken, it will now connect to where this bond of hydrogen has been, where it has removed. And then we shall form oxygen. This one is on carbon atom 1, and now it will be joined on carbon atom 4. So that one is when we shall get an 1,4 glycosidic bond. So when they react, this one is called a condensation reaction. Condensation, why? Because we are losing a water molecule. We are losing a water molecule, so we call it condensation reaction. So the products that we are going to get is going to be this one, and this is now our maltose. Now you can see that here we have carbon 1 and carbon 4 are joined with oxygen atom here. So this bond is what we call a 1,4 glycosidic bond. This is the glycosidic bond joining alpha glucose to another alpha glucose through carbon 1 and carbon 4. So that's why we say that two molecules of glucose can join together to form a molecule of maltose. So here we have formed a molecule of maltose plus our water, which we have lost from here. Glucose and fructose have the same formula, but they differ in the way that hydrogen and oxygen atom are distributed. So here we have glucose molecule, and here we have fructose molecule. The only difference between the two is in the distribution of hydrogen and oxygen atom, the way they are distributed. If you look here, this is carbon atom 1 and this is carbon atom 1. We have carbon atom 2, carbon atom 2 with hydrogen and oxygen. Carbon atom 3, carbon atom 3, the same. Carbon atom 4, carbon atom 4. Carbon atom 5. If you look at carbon atom 5 and look at carbon atom 5 here, then you have carbon atom 6 and carbon atom 6. So you will see that the distribution of oxygen and hydrogen differ. For example, if you look here on carbon atom 1, carbon atom 1 has the hydroxyl, it has hydrogen here, and here it is connected to another carbon, and here it is connected to oxygen. But when you go to carbon atom 1, it has two hydrogens and the hydroxide, then another one is connected to a carbon. So there is a difference in the distribution of hydrogen and oxygen. Sucrose, this one is formed when a molecule of glucose and a molecule of fructose combine. That is when we form our sucrose. So it is formed when a molecule of fructose combines with a molecule of glucose. This is our molecule of glucose, and this is our molecule of fructose. And when they are combining, we are going to use carbon atom 1 of glucose, and here we shall use carbon atom 2 of fructose. 1,2 glycosidic bond will be formed. So here, when you look here, we have the hydroxide and the hydroxide. So we are going to remove this hydroxide with this hydrogen, and we are going to form a water molecule. That is a condensation reaction. So condensation has taken place. And when we lose that one, it means that this bond here is going to be empty. And also this bond between oxygen and hydrogen is empty. So we shall form a bond there. A bond is going to be formed between here and this one here. So we shall form that bond there on this oxygen here and the carbon atom here. And there we shall form a glycosidic bond. Now the two molecules will be joined together by a glycosidic bond, which is 1,2 glycosidic bond. And now it is our sucrose molecule. And this is part of starch molecule. It is part of the starch molecule. Starch and cellulose, these are formed from hundreds of glucose molecules joined to form a long chain. So here we have our long chain of glucose molecules joined together by glycosidic bonds. You're seeing in the middle, that is a glycosidic bond forming very many molecules of glucose joined together, and we form what we call starch molecule. Starch is represented by a single long chain of up to 1,000 glucose units. But in fact, the main components of starch is a branched chain. So when you look here, you will see that this is our starch, and it has branched chains. It can have branched chain. This is amylopectin starch. It has very many glucose molecules or glucose units joined together with very many branches as the main component of starch. So it is made up of very many branches. This is our starch molecule. Proteins are made up of carbon, hydrogen, and oxygen molecules, but with an addition of nitrogen. So our protein is made up of carbon, it's made up of hydrogen, oxygen, but also there is nitrogen molecule. So now when we look here we have two amino acids joined together with a peptide bond. Two amino acids joined together with a peptide bond. And the peptide bond here is formed as a result of condensation, loss of water molecule. So let us try to look at the components or the molecules that make up the protein. So we have said it is made up of carbon, and here we can see there are carbon atoms. We have carbon here in this amino acid. This is carbon and carbon. It is made up of hydrogen. Hydrogen is here. We have hydrogen, hydrogen here. There is oxygen. We have oxygen here. And then there is also an addition of nitrogen. Then we also have nitrogen here. So those are the elements that form our protein. It is made up of carbon, hydrogen, oxygen, and in addition there is also nitrogen. Now we saw that carbohydrates, for them, they are made up of glucose as the simplest unit. Now when it comes to proteins, for the proteins, the simplest unit is called the amino acid. Like the way we have seen here, that the whole of this one is an amino acid here. Here we have the OH attached, and the other side we have hydrogen. So when you look at this one, at this carbon atom here, there is oxygen and there is an OH carboxylic group here. So if you look here, we have this O, which is the same as this one, meaning that it needs now OH to complete the amino acid. Then on this side here there is a bond which was originally attached to hydrogen. So that one forms the amino acid. It has the amino group, and then down it has the carboxylic group. There are about 20 different amino acids. For example, we have glycine, we have alanine, we have valine, there is cysteine, and many other. We have 20 amino acids. The amino acids, glycine combined with valine, cysteine, alanine, glycine, alanine, valine, if they are joined together in that sequence, they would make up a small protein. So amino acids joining with another amino acid, we end up forming a protein. Most of the proteins are very large molecules. The proteins are very large molecules, and they are made up of hundreds of amino acids. For example, here we have our protein here, and this protein, when you look at it, it is made up of very many amino acids. These ones that we are seeing, all these ones are amino acids. So we are saying that most of the proteins are very large molecules because they are made up of hundreds of amino acids. Here we have our protein, which is an enzyme. So if you look at it, it has got very many amino acids. These ones that are amino acids. All those circles that you see there are amino acids which are joined together to form a particular large molecule. Enzymes are proteins which carry out specific reactions in a cell. So we have different enzymes that are in the cells, and they are carrying out specific reactions in the cell, and they are protein in nature. Here we have an enzyme, and this enzyme is acting on a particular substrate here. When you look at it, it is acting on a particular substrate. Now, it is a protein in nature, and it is made up of very many amino acids. Let us try to go through. We say that these enzymes, they carry out specific reactions in the cell. Now, this one is having a substrate molecule situated within the enzyme active site. The enzyme has got an active site where the substrate can fit. This is our enzyme. All of this one here is the enzyme, but this is now the active site where the substrate can fit. 2 of the 6 amino acids which bind with the substrate. These are the 2 amino acid number 40 and 41 are the ones that attach on the substrate, or they form the what we call the active site. Then there is 20, 21, and then we have 3 and 4 amino acid 3, 4, 21, 20, and then we have 40 and 41. They are the ones that form the active site, or the amino acids which bind on the substrate. Now, also as the peptide, the protein is moving around, it forms bonds amongst its amino acid. These bonds, such as the hydrogen bond, which maintain the enzyme shape. Those bonds are the ones that are going to maintain the enzyme shape so that the active site remain intact. And then we have now the amino acid not forming part of the active site. These are other amino acids, but for them they are not forming part of the active site. This part here is what we call the active site. The active site, it is where the enzyme or the substrate fits. Then we have a single polypeptide chain of the 50 amino acid which form the enzyme. Now here we have a polypeptide. It can be a polypeptide of the 50 amino acids that form an enzyme. Proteins make up structure of the cells. It forms the cytoplasm, the nucleus, cell membranes, and enzymes. All of them are made up of proteins. And proteins, they are the ones that make the structures of cell cytoplasm. We saw the cytoplasm and the nucleus. We saw the cell membrane and the enzymes. This one here, which we have here, is our cell membrane. When you look at it, it has got very many proteins. Like, for example, here we have glycoprotein, branching carbohydrate portion of protein which act as an antigen. Here it is also made part of a carbohydrate combined with a protein to form an antigen. Okay, now here we have a glycolipid. This is a glycolipid. We have here cholesterol. We have here a large portion of fat embedded. We have hydrophobic tail of phospholipid molecule. we have integral protein spanning the phospholipid layer. Now this one is also a protein, okay, a protein passing through the phospholipid layer. This one is a protein. We have a peripheral protein lying on the surface. These are peripheral proteins, they are lying on the surface. And then here we have another proteins within the structure or integral with the phospholipids. So you can see that proteins are making up what we call the cell membrane. The whole of this one is a cell membrane, but it is made up of proteins. The way the amino acids are joined up gives a protein molecule a particular shape, which is different from every protein. So the combination of the proteins is going to give the shape of the protein. For example, if you look at this one here, you will see that the way the proteins are joined together, we have a bond here, a bond at cysteine, we have a bond at valine and alanine and glycine, we have a bond at cysteine. So it is going to form a protein with its own shape because of the arrangement of the amino acid. So the amino acids are the ones that are going to give the arrangement of the shape of the protein. Higher temperatures and certain chemicals can cause protein molecule to lose its shape and its properties. So we have said that the amino acid arrangement is the one which is going to give the protein its shape and structure. But we have seen that the amino acids, as they are aligning themselves, they end up forming bonds. And those bonds which are formed are the ones that are giving the shape of the protein. So with temperature, as the temperature increases beyond the optimum temperature, the proteins, the bonds between the protein, the amino acids are going to be broken. And when those bonds are broken, it means that now the protein is going to lose its shape. So when we look at the rate of reaction here and also the temperature, especially when we talk about the enzyme, we have the rate of reaction and then we have the temperature. At the beginning, you can see that the temperature, as the temperature is rising, the rate at which the reaction increases due to increased kinetic energy of the substrate and enzyme molecule. So you can see here the rate is low when the temperatures are low, but as the temperatures increase, the rate also goes on increasing. Why? Because there is increase in kinetic energy of both the substrate and the enzyme. So they are moving very fast, therefore they can come into contact with each other and then the reaction will take place. But again, when the temperatures go on increasing, then the rate again goes on decreasing. So you can see here that the rate of reaction decreases due to denaturing of enzyme molecule. As the temperature increases, the rate of reaction goes on decreasing. Why? When we increase the temperature, the rate of reaction goes on decreasing. Because as we increase the temperature, there are enzymes that we are denaturing. Denaturing means that we are breaking those bonds in the amino acids, and then the enzyme is losing its structure. It's going to lose its structure. So as the temperature increases and it goes beyond the optimum temperature, our optimum temperature it is the temperature at which the enzyme perform to their best or to their maximum. So what is going to happen as you increase more temperature, the rate of reaction is going to reduce. Why? Because the enzymes are being denatured. So this curve that we are seeing is the actual rate of reaction as a result of the combined effect of the two influences. There is this influence of increase in the rate of reaction, and then there is this influence in the decrease in the rate of reaction. So the two, when you get them, you will see that this is the curve that you are going to get. Protein which loses its shape is said to be denatured. This change cannot be reversed. So when a protein loses its shape, it means that we have broken a lot of bonds. And when it loses its shape, we say it has been denatured. The shape has been changed. Therefore, it can no longer work properly, or it has been disfigured that the substrate can no longer fit in the active site. So it has lost its shape. It has been denatured, and that change cannot be reversed. Egg white is a protein, albumin. When it is heated, it is denatured and changes from a clear runny liquid to a white solid. This cannot be reversed. We have seen the eggs. You have ever broken the egg, you will see a yolk, which is always yellow, but there is the albumin, which we call the egg white. That one is made up of protein, and that protein is a runny liquid. It is a liquid which is runny, and when you put it in water which is very hot, what is going to happen is that that runny protein liquid is going to be denatured, and it will turn into a white solid. Now, it cannot be reversed. It cannot be taken back from a solid to back a runny liquid. Why? Because the proteins have been denatured. If an enzyme is denatured, it can no longer carry out its normal function. So when the enzyme is said to be denatured, it means that its active site has been changed. It means that now it can no longer fit in the substrate. And if it can no longer now fit in the substrate, it means that now it is destroyed, or it has been denatured. It can no longer perform a normal function. Let's look at the lipids. The lipids, these are fats and oil. When we say fats and oils, all of them are lipids. What is the difference then between fats and oils? Fats, these are lipids that are solid at room temperature. And oils, these are lipids which are liquid at room temperature. All of them are lipids. They are made up from glycerol and fatty acid. They are made up from glycerol and fatty acid. So here we have our lipid here. It is made up of a glycerol. These are glycerols we have here. That is a glycerol molecule. And this side we have the fatty acid molecules. 3 fatty acid molecules combine with 1 glycerol molecule to form a lipid. So these are our lipids, which are fats and oils. They are made up of glycerol and then the fatty acids. For example, fatty acids, we have stearic acid, we have oleic acid, and we have palmitic acid. So these are some examples of the fatty acids. Now you can find that on your lipid here, one glycerol may combine with the stearic acid. This can be stearic acid, stearic acid, stearic acid. Or it can be here having stearic acid, and here we have oleic acid, and here we have palmitic acid. So these are the fatty acids: stearic acid, oleic acid, palmitic acid. But we have only one glycerol combining with 3 fatty acids. The fatty acids in a lipid may be all the same. So here we are having our lipid, and we are saying that all the fatty acid 1, fatty acid 2, fatty acid 3, they may be the same fatty acid. In olive oil, for example, the fatty acid is oleic acid. So in olive oil, you find that fatty acid 1 is oleic acid. Fatty acid 2 is still oleic acid. And even fatty acid 3 is oleic acid. So 3 oleic acids are joined with 1 glycerol. In addition to proteins, carbohydrates, lipids, cytoplasm contains salts and water. So we are looking at some of the biological molecules. We have proteins, carbohydrates, lipids that we find in the cytoplasm or in the cell. But in addition also to those biological molecules, we may have also salts and water. So here we have our water molecules, and the one water molecule here has a connection with another water molecule, and these connections are what we call the hydrogen bonds, what we are calling the hydrogen bonds. That is the connection between the water molecules. Water makes up the bulk of the cytoplasm. So as we look at the cell and then we'll say the cytoplasm, we know where the cytoplasm is. That point where we call the cytoplasm, it is mainly made up of water, though it has other components. Mitochondria, we have Golgi bodies, we have virtual starch granules, and so on. All of those ones are in the cytoplasm, but the bulk of it is water. All the chemical reactions in the cytoplasm, they take place in solution, and that is in water. So we saw that most of the chemical reactions in the cell, they take place in the cytoplasm. And these chemical reactions, they take place in solution, and this solution is water. Water itself takes part in many of these chemical reactions. Now, in addition to the cytoplasm being made up of a higher percentage of water, also these chemical reactions that are taking place in the cytoplasm, they involve water. Water takes part in this chemical reaction. In photosynthesis, water is going to react with carbon dioxide to form glucose. That is, in photosynthesis, we see that water, 6 molecules of water, they react with 6 molecules of carbon dioxide, and we end up getting 1 molecule of glucose and 6 molecules of oxygen. So you can see that water is taking part in the reaction which is taking place in the cell, and that is an example given out in photosynthesis. Salts, for example, sodium, potassium, we have calcium, and many others are very important in these reactions. We have very many salts that take part in these chemical reactions that take place in the cell. Enzymes are special proteins. Now we want to look at the enzymes. These enzymes, we are saying they are special proteins. We have seen earlier that the enzymes are made up of small units, and those small units are the ones that we call the amino acid. Amino acids are the ones that join together to form a protein, and an enzyme is a protein in nature. They are present in the cytoplasm of all cells. Enzymes are present in the cytoplasm of all cells. Why? Because we said in the cytoplasm, that's where most of the chemical reactions take place. And this chemical reaction, they take place in the presence of enzymes. Therefore, enzymes are also in the cytoplasm. They help to speed up the chemical reactions in the cell. So the main functions of the enzyme is to speed up these chemical reactions that are taking place in the cell. There are hundreds of different enzymes, but each enzyme speeds up only one kind of a reaction. So we have very many enzymes that we find in the cytoplasm, but each of these enzymes speeds up only one kind of reaction, meaning that these hundreds of different enzymes, they take part in different kind of reaction, and their work is to speed up the rate of the chemical reactions. For example, glucose and fructose might join up slowly to form sucrose. So here we have our glucose, alpha glucose, and we have beta fructose here. They are combining up together, through condensation, they are going to lose water. And when they lose water, we shall form a glycosidic bond. So here we are going to form our product, which is sucrose. Glucose has joined or combined with fructose via a 1 to 2 glycosidic bond to form glucose. So this reaction may take place very slowly. With the right enzyme present, the reaction happens faster. So when we have a reaction like this one, which is a condensation reaction, and it is taking place in the absence of an enzyme, the product is going to be obtained, but at a slow rate. But when we have the right enzyme, that right enzyme is going to speed up the rate of the reaction, and the product is going to be formed very fast. So the enzyme, their main work is to speed up the rate of the reaction. This reaction would take place, but it would take place at a very slow pace. But when we have the right enzyme, it will take place at a faster rate. Enzymes cannot alter the end product of a reaction. The enzymes cannot alter the end product of a reaction. For example, if we have been having our enzyme here, which has speeded up this reaction, and we have got sucrose as our end product, then this enzyme still now cannot again change this product into another form. So they can only speed up the reaction which would take place without the enzyme, but too slow to support life. So we can see that the enzyme, they can only speed up the reaction which would take place without the enzyme, but too slow to support life, which means that the enzymes are speeding up a reaction, and that reaction could take place without enzyme. But the only problem would be that the reaction would be too slow, that it would not support life. So to support life, that's when the enzyme comes in to support this reaction to take place very fast, so that that reaction can support life. Enzymes are large protein molecules. Enzymes are large protein molecules. We have seen that enzymes are made up of amino acids which join together to form a protein, and that protein now can form what we call an enzyme. For example, if we look at our enzyme here, this enzyme is made up of 50 amino acids. All these 50 amino acids, they join up together. And when they join up together, but there are only very few amino acids that are going to participate in the reaction. For example, if you look here, this is our substrate. Substrate molecule situated within the enzyme's active site. So the whole of this enzyme, it has a point where the substrate fit. That point where the substrate fit is what we call the active site. And this active site has got about 6 amino acids which bind to the substrate. So you have here our protein, which is the enzyme, is made up of 50 amino acids. But there are only 6 amino acids which are forming the active site. Those are the amino acids that are binding with the substrate. For example, here you can see amino acid 3 and 4, they are binding on the substrate. Then we have 21 and 20, they are binding on the substrate. Then we have 40 and 41. So there are 6 out of the 50 amino acids which are binding the substrate, or which form the active site. Then we have also these other amino acids not forming the part of the active site. There are also these other ones, these other amino acids, but for them they are not taking part in the forming of the active site, but they are needed for that particular enzyme. So they will fold and then they will form bonds such as hydrogen bond, which maintain the enzyme shape. These amino acids are needed for that particular enzyme, and then they are needed to form bonds, which bonds are going to form the active site of the enzyme. Like all proteins, each enzyme molecule has a particular shape. So that's what we have been looking at. That like all protein, each enzyme that we have in the body has got its particular shape. So when you look at this protein here, which is an enzyme, it has its particular shape because it is working on a particular substrate. So it forms a shape where that substrate can fit, and it will only work on this type of substrate because this type of substrate is the only substrate that can fit in the active site. So it has only this particular shape to work on this particular substrate. This shape determines which chemical reaction the enzyme can speed up. So if we look at our enzyme here, just look at its active site. When you look at its active site, it can only fit in the shape of this substrate, meaning that the chemical reaction it is going to be involved in, or the chemical reaction this enzyme is going to speed up, should be determined by this shape. Any other substrate that has got a different shape than this one will not be worked on by this enzyme. So that's why we are saying that this shape determines the chemical reaction the enzyme can speed up. Speeding up a reaction, the enzyme combines temporarily with the substrate it is acting on. So when the enzyme is speeding up a chemical reaction, what is going to happen? The enzyme is going to combine with the substrate for a short period of time. It will combine with the substrate so that it facilitates the faster rate of reaction of that reaction. Any substance an enzyme acts on is called a substrate. the substance acted on by the enzyme is what we call a substrate. This substance that we have here, that is being acted on by the enzyme, is the substance that we are calling a substrate. Enzyme and a substrate. The substance or the substrate molecules fit the shape of the enzyme. So, if this enzyme is working on this substrate should fit into the enzyme. It should fit into the active site of the enzyme so that it can be acted upon by this enzyme. Let us look at how the enzyme is working. Now, the substrate molecule will fit in the shape of the enzyme. So we have here our enzyme. This is our enzyme, and it has got the active site. This one is our active site. We have the active site. This point here is the active site, which is on the enzyme. This active site on the enzyme, it is where the substrate is going to fit. That is the point where the substrate is going to fit. Now here we have our substrate A and we have substrate B. They are going to fit into our enzyme. So the blue oval represents an extremely simplified form, an enzyme molecule with a particular shape. So here we have our enzyme with a particular shape. You can see this particular shape where the substrate is going to fit. The slots in its side represents the active site into which the substrate will fit. So these are our slots. These are our slots. These are our slots where the enzymes or the substrates are going to fit. Those are our slots on the side of the enzyme, which we call the active sites. They are the active site. And in these active sites, it is where the substrate is going to fit. The pink shape represents the substrate molecule. So our pink shape here, this shape here, this is a substrate molecule, and this one is also a substrate molecules. They are the ones that are going to fit in the active sites. The substrates will combine temporarily with the enzymes. Now we can see that our substrates here, now they have combined with the enzyme. Temporarily, they combine with the what? With the enzyme using the active site. They have combined with the enzyme by fitting into the active site. Enzymes join the substrate together. Now our enzyme, what it is going to do is to join these two substrate together. Each one came alone, but now the enzyme is going to help them to join them together so that they are joined together. They form now one product. That is the work of the enzyme. It speeds up that reaction so that the two substrates can now join together very fast. The reaction between the enzyme and the substrate is sometimes described as the lock and key mechanism. It is referred to as a lock and key mechanism. Now, when you look here, our substrate is going to act as a key. It is like a key. And the enzyme is acting as a lock. So you bring the key, you push it into a lock. So this reaction, whereby the enzyme is combining with the substrate, is most cases described as lock and key mechanism. So an enzyme is involved in combining substrates to create a larger molecule. So here you can see we have created a larger molecule. Now the enzyme's work was to help speed up this chemical reaction so that these two substrates can combine together very fast. So here we are saying the new compound released by the enzyme, and the enzyme is unchanged and is ready for the next reaction. So that's why we are saying that an enzyme is involved in combining substrates to create larger molecules. For example, enzymes could build up sucrose from molecules of glucose and fructose. So an enzyme can help to combine or to join glucose with fructose to form a larger molecule, which is sucrose. Enzymes are named according to the substrate on which they act, and the names all end in ASE. So when the enzyme is acting on, for example, joining or formation of sucrose, the name of the enzyme is going to have the name of the product. For example, the product is sucrose. So on that sucrose, we shall remove OSE and we add ASE. So that enzyme now will be known as sucrase. Sucrase. It is named according to the substrate on which they act. Many enzymes which act on proteins are called collectively proteinase. 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 Those are enzymes which act on proteins. In the next sequence, Schlol shows the enzyme can help to break a large molecule into a smaller molecule. Now let us look at how enzymes are going now to break large molecules into simple or smaller molecules. For example, sucrase. sucrase acts on sucrose to produce fructose and glucose. And maltase enzyme will act on maltose to produce glucose. So let us look here on our sucrose molecule here. We have our sucrose molecule and we are going to break it down into glucose and fructose. And here we are going to hydrolyze the sucrose. Hydrolysis means we are adding a water molecule. We are going to add a water molecule. That addition of water molecule is what we call hydrolysis. And this reaction can take place without the presence of an enzyme. But only that now it will be very slow. So to speed up the hydrolysis process, we are now going to use an enzyme sucrase. Sucrase is the one which is going to bind the sucrose and speed up the hydrolysis process. When water is provided, it's going to break the 1,2 glycosidic bond. It will be broken and we shall add in the water molecule there. When we add in the water molecule, the bond is going to break. Now this one will go on its own and this one will go on its own. That's why we are ending up forming glucose. And this side we form fructose. So what has helped us for this reaction to take place very fast? It is the presence of sucrase enzyme. Now, a breaking down reaction. Let's look at a breaking down reaction. Now, let us first look here. We see that here we have, this is called the active site of the enzyme. Now, all of us, we know that the active site, it is where the substrate are going to fit. And now here we have our substrate here. The shape of the substrate molecule is going to fit into the enzyme shape. So it is going to fit into the enzyme shape there. The enzyme could be, for example, sucrase. Our enzyme is sucrase. And the substrate is the sugar sucrose. So we are assuming that this enzyme here is enzyme sucrase. And enzyme sucrase is going to work on our substrate here, and our substrate here is substrate sucrose. It is going to be acted on by the enzyme sucrase. The enzyme will split sucrose molecule into glucose and fructose. So the purpose of the enzyme is to split this substrate, which is sucrose, into glucose and then fructose. That is the work of the enzyme. This reaction is a hydrolysis reaction, and it is going to break down sucrose into glucose and fructose. But we are using an enzyme sucrase to speed up that chemical reaction so that sucrose is broken down into fructose and glucose very fast. The substrate combines temporarily with the enzyme. So here we are having our substrate is already now combined with the enzyme, but it is going to be temporarily for a shorter period of time. So the enzyme will break down at this point. The enzyme is going to break down at this point. Now the substrate splits and separates from the enzyme. So when the substrate has been now split into two parts, it will separate from the enzyme, meaning that now the enzyme has finished its action and it is ready to perform another reaction. The final breakdown product, Here you can see that the enzyme can carry out thousands of reactions like this one in a second without itself being used up. So we end up getting our end products, many of them acted upon by our enzyme. And the enzyme, after breaking the substrate into products, it is ready to perform another reaction. And it can do thousands of reactions like this in a second. And for it, its shape is not going to change, meaning that the enzymes are not changed by the reactions they carry out. So the enzymes, they belong to a class of chemicals called catalysts. Enzymes, they belong to a class of chemicals called the catalysts. A catalyst can speed up a chemical reaction without being used. So an enzyme is in the class of chemicals called catalysts. And these catalysts, they speed up the rate of a chemical reaction and they are not used up, meaning they are not changed by the reaction they take part in. A catalytic converter in a car exhaust system speeds up the conversion of carbon monoxide, oxides of nitrogen, and unburnt fuel to relatively harmless compound. We have other types of catalysts that are found in vehicles or catalytic converters in exhaust system. These ones, they speed up in conversion of carbon monoxide, oxides of nitrogen, and unburnt fuels into products which are less harmful or harmless. The catalysts may be platinum, it can be chromium, or any other metal. Those ones may be catalysts. But enzymes, they work a great deal faster than the man-made catalyst and at a much lower temperatures. So the enzymes, for them, they work faster than these inorganic catalysts, which are man-made. And for the enzymes, they work at a lower temperatures compared to the man-made catalysts. Let's now look at the properties of enzymes. Properties of enzymes. Enzymes can act on only one type of a substrate. An enzyme can act on only one type of a substrate. Let us try to look at these enzymes that we have. This is our enzymes, and look at its active site. The active site, it's where the substrate fits, meaning that this enzyme works on only substrate which have got this shape which can fit in the active site. But when you look at this substrate here, the shape of this substrate cannot fit here. Therefore, this enzyme cannot work on this substrate. The same applies on this one. When you look at this shape here, this one is the shape of the enzyme. And look at the shape of the substrate. This substrate cannot enter into these active sites because it cannot fit there. Therefore, it means that this enzyme cannot work on this substrate. But when you look at this enzyme here, if you look at this enzyme, it can work on this substrate. This substrate can fit into this enzyme. If you look at the shape of the substrate, you can see that that shape of the substrate can enter into this enzyme. At the same time, this enzyme here can work on this substrate, because when you look at the shape of this substrate, you can see that it can enter into this enzyme. So that's what we are saying, that enzyme can only work on one type of a substrate. These substrates cannot combine with these enzymes. So you can see that this substrate here cannot combine with these enzymes. By all means, it cannot combine with this enzyme. And this substrate here cannot combine with this enzyme. So these ones cannot work on this substrate because their active sites are not corresponding. They always produce the same product. They are always producing the same end product. So if we have this enzyme and is working on this substrate, it will always produce the same products. We have seen that our enzyme can work on these substrates, thousands of them per second, but it will end up always producing the same product. It cannot change and produce other products. Although they take part in the reaction, they are not used up. So we are seeing that our enzymes are taking part in millions of reactions, but for them, they don't change their shape, meaning that they are not used up. So that means that an enzyme can work on millions of reactions. One reaction, one enzyme working on millions of substrates. Because enzymes are protein, they are denatured by heat or some chemicals. We saw that the enzymes are made up of proteins, and the proteins are made up of amino acids. And these amino acids, they form bonds and join together. And when they form these bonds and form hydrogen bonds, these hydrogen bonds, they maintain the shape of the enzyme or the shape of the active site. But when you apply a lot of heat, that heat is going to break the bonds. And when you break the bonds, now you are going to change the shape of the enzyme. And when the shape of the enzyme is changed, we say the enzyme has been denatured. How denaturation comes about? When there is change in the shape of the active site. And when you change the shape of the active site, it means that now the substrate this enzyme works on can no longer fit into the active site. Denaturing involves change of shape in the enzyme molecule so that it cannot now combine with the substrate. So now if you look at our enzyme here, it has been denatured. And when it has been denatured, the active site shape has changed. Now this shape of the substrate cannot fit there. If you look at this shape of the substrate, it cannot enter here. And if this one cannot enter here, therefore this one also cannot enter there. Therefore, the enzyme now cannot work on this substrate. Individual enzymes, they work best at a particular temperature and pH. So our enzymes, they work at a particular temperature and a particular pH. When the temperatures are low, the rate of reactions are also low. But as we increase the temperature, we shall see that as the temperature increases, if you extrapolate, you will see also the rate of reaction is increasing. When you keep on increasing the temperature, you will see that now also the rate of reaction is going to increase by increasing the temperature up to when we reach what we call the optimum temperature. At the optimum temperature, this is where the enzyme acts very well, the maximum temperature at which the enzyme works best. And it is the one that gives us the maximum product. But when the temperatures increase beyond the optimum temperature, then the rate of reaction is now going to start reducing. It goes on now going where? Downwards. Why? Because now it means that our enzymes are being denatured. So different enzymes, they work best at particular temperatures. Some enzymes work best at lower temp. All enzymes work best at the optimum temperature, which is ranging between 40 and 45. And in pH, you'll find that in pH, some enzymes work better in lower pH, others work better in little pH, others work best in higher pH. So each individual enzyme works best in its particular temperature and the pH. Individual enzymes, as we have already seen, individual enzymes, they work best in a particular pH, which is acidity or alkalinity. Now, when you look at our graph here, you are going to see that this is the rate of reaction and this is the pH. We have different enzymes here. And these different enzymes, they are working at different pH. So you can see here we have pepsin. Pepsin is working best at the pH of 2. And when you look at sucrase, sucrase works best at the pH of 4.5. When it comes to salivary amylase, it is working best at the pH of 7. And then agnase works best at the pH of 10. So each individual enzyme works best at a particular pH. So if you look at pepsin here, pepsin works in acidic pH. But when you look at agnase, agnase works in alkaline pH. That's when it gives us the maximum rate of reaction. So a glucose molecule combines with the active site on the enzyme. So here we have, these are our glucose molecules, and here we have our enzyme. Now this enzyme has got active sites. These are our active sites. The active sites are these ones here on our enzyme, and these glucose molecules will combine with the active site. So now you can see here the glucose molecule has come and it has attached itself on the active site. A region of the active site is still available. Now we have here our enzyme. If you look at our enzyme, one of the active site has been combined with the substrate, but we still have another active site which is still available. Now this active site, it's where another molecule of glucose will come and attach itself, and then we shall have the hydrolysis or the condensation reaction taking place. So one end of a growing starch molecule will combine with the glucose molecule at the active site. One end of a glucose molecule will combine with the active site. Now this one is part of our starch molecule. This is part of our starch molecule, which we want, on this starch molecule, we want to add more glucose molecule on this starch molecule. Now we have here our enzyme, which has already attached to a glucose molecule. Now we want to add this glucose molecule to this starch molecule. So this starch molecule is going to use one end of it to come and attach itself on this active site. Then we shall combine and form 4 glucose molecules attached together to form a bigger starch molecule. So here we can see that we have brought the other part of the starch molecule, and when it comes, it combines with this free active site, and then we combine this glucose molecule to this group of starch molecule. So the growing starch molecule will break free from the enzyme, which is now free to repeat the reaction. Now we have our enzyme here, which has taken part in the process of combining this bigger molecule of starch molecule. Now what is going to happen is that this growing starch molecule will break away. It will break away from the enzyme, and the enzyme now will continue its cycle of combining more other glucose molecules to the growing starch molecule. So now you can see, now our starch molecule has moved away from the glucose molecule, and now the enzyme may carry out reactions like this one 1,000 times in a second without being used up itself. So it has worked on this molecule here, and it has increased the length of the molecule. Now this molecule will go away, and now the enzyme can repeat the same reaction and produce more molecules. Thank you very much for participating in chemical reaction. Please do like, share, and subscribe.